All right. Good. Then, good morning, everyone. My name is Oliver Kohlbacher, and together with the three gentlemen introduced already, we hope to have some fun for the next 2.5 days and guide you through the mystery that is OpenMS, and also give you some solid background on what is going on in mass spectrometry-based proteomics and metabolomics. So I am a professor of applied bioinformatics, as you can see, at the University of Tübingen, where I have been teaching courses on mass spectrometry and related topics for well over a decade. And I want to start this morning off by giving you some background on the fundamentals of targeted proteomics, of non-targeted proteomics, metabolomics. So let me start with a couple of questions. Who has ever measured mass spectra in a lab? Who? Then some of the things I'm going to say will be perhaps a bit boring, because you might have done that already, but please bear with me. It's going to be over in less than an hour, and I promise, even if you tried some of these things, actually acquired data on your own, you might find some of the perspective a bit slightly different one, because we are going to talk about computational mass spectrometry. And I will start by sort of defining the overall area, and then we will zoom into a few particular topics. You'll see that there is always a mix of lecture part and Timo and Oliver are going to hand out the materials. We brought some USBs. You have those already, and we will walk you through that. There are also tutorials that you can then use, and they will show you step by step how this goes. Now, you can imagine there are people in the room that have people that are, and it's going to be mixed. So we designed this in a way that have eventually is happening at those lectures, but that doesn't mean you can make your way through the hands-on at different speeds or with different priorities. We will explain that a bit later on. We will then have another lecture after lunch on database search, so really the fundamentals of mass spectrometry, why we actually know what proteins are in there. Then we will try that ourselves, and then I will talk briefly about what's called false discovery rate estimation. What does that mean? How does it work? So basically, how can we trust the data that we generate? And that is another interesting topic that is often not discussed at the depth it deserves. I was earlier we're not entirely sure. Just sure we can't do it in this room. We're not entirely sure whether we have a room. So it might be that we move that to tomorrow morning at 8, where we also have a slot for it. So by this afternoon, I'll let you know. Then tomorrow we will talk. Today we're talking about qualitative proteomics. So just figuring out what is there. Tomorrow we're going to talk a bit more about how much of that is there. And in the morning we will do that for proteomics, so we will discuss label frequency. On that, who is working in proteomics? Who is working? Okay, anybody working in both? Great, that's the way to. Metabolomics data now, or on proteomics data right now? Are essential, and it might also give you an idea on how to continue with that. And with metabolomics, I mean also other small molecules. Let me start the brief introduction to proteomics and metabolomics, and I'll actually start by introducing something completely different, but hopefully related, and that's systems biology, which is usually the reason for doing proteomics and metabolomics. I give key ideas of what we do there, and then we will start discussing LCMS, which is the key technology that we are really going to use. where we are going to use OpenMS. I'm going to explain the philosophy of it, use some pointers on how to get started with it. Yes, please. No. Well, the microphone is just for the recording. So, but I can try to speak up a bit, and you just complain to me I'm not loud enough. And just throw stuff at me or shout or raise your hand or whatever. I would appreciate this more interactive. An hour and a half. If you throw in questions, just please don't hesitate. Just shout, raise your hand or whatever, and I'll sort of try and motivate some questions at some point. So let's get started with something that everyone in the room should have seen or should have heard of, and that's a central dogma of molecular biology. So it's kind of interesting in case you haven't seen that here. This is from Francis Crick, October 1956, where he sketched his ideas on protein synthesis. And fundamentally, the idea is rather trivial and known to you. We start with DNA, go to RNA, and protein, and these arrows basically indicate the flow of information. So DNA is the blueprint for what is then transcribed into messenger RNA, which is then translated into proteins. Why is that interesting? Because it basically on the proteome as well. We just need to follow these arrows backwards until we arrive at the proteome. And that is why genome sequencing, in particular the sequencing of the human genome, but if you work in different organisms than these as well, has been so essential also for proteomics, because it basically is blueprint for all the genes, all the proteins one can discuss, how one defines a gene, how one defines a protein anyhow. But with the publication in February 2000, the first time when Nature and Science actually appeared on the same day, and they each claimed to have the human genome, but they both had different genomes, of course. It was a very interesting event, press conference in the White House, and they announced we have the human genome. Of course they didn't. Do you know how many genes they published back then? So how many? Yes. Oh. Well, you should know. Yes. Well, didn't see you in the corner. Good. So the number was overestimated. How many human genes are there that are annotated at the moment? Yeah, 20,301. The last time I checked, this number keeps on going up. And that has to do with the fact that it's not that easy to really figure out what is a gene, and we would need to have a definition of that in the first place. But if we assume that a reference genome gives us all the sequences of all the proteins that should be synthesizable, then it is clear that this genomic information is an informed foundation for all the other, what we then call post-genomic methods, because they rely on the information that we have. And we should probably talk a bit about this whole omics thing. When you look that up, what does ohm really mean? Then there's actually an entry in the Oxford English Dictionary that says, in cell biology, molecular biology, forming nouns with a sense, all of the specified constituents of a cell considered collectively or in total, as plastidome, plastome, vacuome, and so on. So it's essentially saying we're doing something on a large scale or on the genome-wide scale. And where the geneticists just got an awful lot of money by deciding to call themselves genomicists and doing genomics rather than genetics, all the protein analytics guys said, well, they pulled off the trick. Let's stop doing protein analysis and rather do proteomics. And that trick tends to work very reliably. So there has been a whole flurry of omicses there is actually a list, omics.org, where you can find really strange things like the alignment tome, which is the set of all alignments. There is the animalome, probably what you used to call it. And there is the antiome, of which I'm a part of, the totality of people who object to propagation of ohms. So fundamentally, this is just bullshitting your way into a successful grant proposal, and that's why we all do it. But when you think about it, then these are the same biomolecules that have been essential for molecular biology for a long, long time. So we are molecules, perhaps lipids and sugars. These are the fundamental types, and all the other stuff doesn't really matter. And if we are looking at the DNA level at the whole genome, so the set of all genes that are encoded, we are looking at the transcriptome, which are all the transcripts that a cell can generate based on this genome, and that's on the mRNA level. We are looking at all the proteins that can be generated out of these transcripts, and we're looking at the metabolome, which are all the small molecules which are generated, catalyzed, converted by these proteins. That's sort of the simple view on that. Now, a quick question. How large is the proteome? Let's stick with human. Yeah? So let's assume we are looking at human. How many human proteins are there? Yes.
It's very different. It's not fixed something. It depends on which.
The max.
Around 60,000.
60,000? More? 100,000? More? Less? How realistic is that? So, has anybody ever worked with the human immune system? But you have one, right? How many different antibody sequences does each of you generate? Immunoglobulins. Well, they all have different sequences, and it's probably on the order of 10 to the 10. So it depends on how you define protein, yeah, because without going into details of immunology, these antibody sequences are generated by genomic rearrangements, by shuffling. So they are not really all of these proteins are encoded. Also, we have post-translational modification. So it depends on how you define it. If you look at splice variants only, if you say all the different isoforms of a protein, then you probably end up at, give or take, 80, 100,000 different proteins. Each of those can be degraded, can be truncated, can be modified in various positions, and that gives rise to a very large proteome. So we really don't know how large that number is, but I think it's a good idea to believe that there are hundreds of thousands at least. So how can we know how many of these proteins, how many of these genes are there? What is the size of this whole thing? We basically have been able to do omics because there are two fundamental technological breakthroughs that enable that. And on the one hand, that has been next generation sequencing, or as we should probably call it now, massively parallel sequencing, because there have been many generations. So this is like next generation by now. And that is able to more sequence everything that is encoded by nucleic acids. So the genome, the epigenome, the transcriptome, the RNome, all of that can be sequenced. And there's a fundamental difference between this part of the omes and the right-hand side, which is the proteome, interactome, metabolome, lipidome. These are usually measured using mass spectrometry. Why is doing mass spectrometry so much harder than sequencing? That's something the sequencing guys do not like to hear, but it's true. What's the fundamental difference between? Exactly, there's no amplification. You know, the genome has a dynamic range of, well, 0. You have the same number of copies of everything, and you can amplify it easily using PCR. But on the proteome, you have probably 12 orders of magnitude dynamic range when it comes to the sheer copy number of the individual proteins, and that makes it a lot harder, and you can't amplify it. There's no PCR for proteins. Nevertheless, sequencing is kind of useful for proteomics because we will see, as we'll see in a minute, it's fundamentally the only way to figure out the sequence of these proteins. Sequencing has gotten a lot cheaper. I'm sure you have seen this figure at some point before. This is regularly updated by the NIH. It's basically the cost per genome as a function of time. And this, note that this here is log scale, 100 million, 10 million, 1 million. And this line here is Moore's law. So basically, compute gets cheaper exponentially. And this straight line in this log plot is an exponential decay of the cost. But the cost of sequencing is dropping a lot faster than exponentially. And that is why it becomes very easy to access the data. It used to be that sequencing one genome was prohibitively expensive. And that is also why proteomics, metabolomics were competitive in that area. But by now, sequencing a patient, sequencing a human genome, what does it cost? Well, it's 300, 400 bucks. Yeah, that's all it costs. So it's basically gotten very cheap. It's price-wise, it's on the same level as proteomics, metabolomics. So once we had this human genome, the next step would have been to go for the human proteome, go for the human metabolome. But for reasons that we just discussed, it's not that easy to know what is really part of this human proteome. But there was a publication in 2014, so 13 years after the human genome, the human proteome made it on the Nature cover. 2 papers, actually, that claim 90% coverage. And we can re-examine this claim at some point later when we've discussed the topic of false discovery rates. Fundamentally, they said that they now have the majority of these genes sequenced. Does that help us in any way? Not really, because we can use that as a reference, but fundamentally, as we will see in the next lecture, we are still going back to the genome to really figure out what is in a sample. So let's go one step back now that I've introduced genomes and proteomes. I would like to discuss why we do that and why these high throughput techniques are different from what we have been classically doing in the lab. Because people have been analyzing nucleotides for a long time. People have been analyzing proteins in the lab for a long time. None of that is new. People have been doing mass spectrometry for a long time. But we're doing it on a different scale now. So we can really sequence all of DNA, all of the transcripts. We can look at all the proteins. We can look at all the metabolites. And we can also look at all the protein-protein interactions, hopefully. So when we're talking about omics data, we're really talking about a broad range of data. But you might know the no free lunch theorem. There is no free lunch, not even today. You have to pay for that somehow. And you pay for that by having reduced reliability of the data. So you get a lot of data, but it's helpful. And having more data means that the individual data point might be less reliable than if you do a gel in replicate and really look at only one protein. You might get a more reliable number than doing it for 5,000 proteins in parallel. So we have 2 types of data. One I would like to call classical. You know, the classical biochemical assay that gave you one number, and that was high reliability. They tend to be low throughput, whereas omics data is always high throughput by definition. By high throughput, I don't mean many samples per day, but I mean many measurements per sample, per run, per injection, whatever you want to call that. Classical data is dimensional. In the simplest case, it's just one number, 55 arbitrary units, whatever that is. With omics, you tend to have high dimensional data with noise low accuracy. And that means the analysis of data is often straightforward. You have 3 experiments, you do perhaps a t-test, and you're done. But many people struggle with analyzing omics data, because there you really need to have statistics, you need to have software, and that's probably why most of you are here anyhow. So when we look at these different omics layers, to me, that reminds me of this cover of a book. Who has seen that book? Yeah, it's by Douglas Hofstadter, Gödel, Escher, Bach. In case you haven't read it, do it. It's an old book, and he constructed what he called wooden, and they're carved in a way that when you shine the light from different direction, the shadow actually spells out different letters. So from the top it's a B, from the side it's an E, and from the other side it's a G. Gödel, Escher, Bach. And why these gentlemen end up in one book, that is for you to explore. So fundamentally, this illustrates how looking at a cell with genomics, with proteomics, with metabolomics reveals completely different pictures, because it's an entire different set of the biomolecules that are contained in the cell. Some things you can see in the genome. If there is a genomic aberration, then you might find that on a proteome level, but why go there? If there are things that are affected only on a metabolite level, you might just not see it on the genome level. So actually looking at biomolecules in parallel is probably the only way to figure out what ask different questions of them. Genomics is basically the full potential of the cell. What can a cell do? But very little of that is ever being realized at one particular time in space. In transcriptomics, the transcripts, it's often misunderstood. People believe that if a gene is turned on, or if a transcript is there, then that also means there has to be the protein. No, it doesn't mean that. One is actually the first derivative of the other, because the number of proteins being synthesized. It doesn't mean that it's actually there, because it's all being degraded. So it's telling you a very different story, and that is why the correlation between transcript and protein is often very poor. 0.4, 0.3, depends on what you're looking at. Proteomics thus tells you what's currently there, not what's currently being produced. It tells you what is really active in the cell. And metabolomics tells you what is actually going on a small molecule level, because these proteins, if they're sitting around, that doesn't mean that they're actually producing something. So all 4 layers highlighted here give you a completely different semantic, different meaning of what a cell is currently doing. In an ideal world, when we're talking about systems biology, really understanding the system, would have insights on all these levels. You would know what the cell is able to do by knowing the genome, would be able to tell what it's trying to do by seeing what it has just turned on the transcript level. You would be able to see what it is actually able to do by seeing the molecular machines that are currently active. That's the proteome level. And you would see what it has produced, what it is producing on the small molecule level. From the way I worded that, you can already figure out that all of that is dynamic. And the dynamicity, the dynamics of that actually goes down as you go down this level. The genome is more or less static. You can argue about that. What's dynamic about the genome? You can have mutations. What else? Excuse me? Transgenic, yeah, but let's assume we're not fiddling with the cell. I mean, then everything can happen. But there are other things that can happen. Well, you can also turn genes on and off on the epigenetic level. That is something that can happen. We learn more and more about modifications of DNA that are essential. You know, methylation, of course, but there are other modifications. So there is some dynamics there, but the transcriptome much more dynamic because you realize genes are constantly being turned on, turned off. This is part of the regulatory process. But the proteome had for. So there are very different also scales associated with these different levels. What we're trying to do in bioinformatics is now to analyze these different sets, and that is what everyone is going for, this holy grail. You have basically all the biomolecules at arbitrary precision, at arbitrary temporal resolution, and then you try to bring it together and make sense of that. That's not trivial. Starting with the measurement or the integration. We know that we have all these pathways, we have these networks that are in the textbook, that are in databases. And what we really need to do is we need to map all these measurements that we had, map the transcripts onto the networks, map the interactions, map the metabolites and regulations, and then somehow bring them together. That's something we will not discuss in this particular course at length because it's complicated. This is an introductory and rather simple course. But fundamentally, that's why you need us bioinformaticians, and we're not going to go away for a long time. We really need to have computational methods that analyze the data, turn it into meaningful units, meaningful quantities, and then try to build models on that. Now, why is that difficult? You could argue that is not so trivial. Once we have all the proteins, what's the real issue there? There are challenges on different levels. There's a semantic integration. You might argue until it fits a new format, then you know that this is actually a non-trivial challenge. And there, one of the big issues is that people are not standardizing methods, are not standardizing file formats. We will get back to that as well. There are also more fundamental things, ambiguities. No? If we cannot easily match protein, if we have different databases, we have different sequences, if we have human genomic variation, then you can imagine that there are many pitfalls that you can find. Oftentimes we also have a lack of data. So proteomics claims to give you the full proteome. Is that true? So how many proteins can you actually see when you do proteomics? Those of you who have done proteomics. Thousands? Yeah. So you need to push really hard to see more than 10,000 proteins. Whether you really see them, if you get these high numbers, that's a different story that we can discuss. But do you really see everything about the protein? Well, of course not. So the coverage of the proteome and also the coverage of the sequence of each individual protein is small. And we should be honest and say, well, for the metabolome, we have basically no clue what we're covering because we just don't know what's there. We just keep on finding more and more. We can discuss whether that is biologically meaningful or not. But this claim that we see everything that is basically possible on the genome level, but all the other levels, it becomes increasingly hard to see that. Then we have different scales. How fast is a metabolic reaction? What's the typical time scale for one molecule to be converted into another? From minutes to days. Then you might be in trouble. That would mean you have a very slow metabolism and probably be starving at this point. No, it's a lot faster than that. Other ideas? Chemical reaction. We're really talking chemistry here. Yeah? So a chemical reaction takes 10 to the minus 12, 10 to the minus 14 seconds. So that's really, really fast. How about gene regulation? How long does it take to turn on a gene? few minutes. Yeah? Things are never, in biology, things are never trivial, but yeah, we're talking usually minutes to hours. It depends a bit on that. Where's signaling in that scale? And why are we discussing this here? Well, we discuss that because this is essential to make sure that you really understand what you're getting in terms of data. I've been working with collaboration partners who generated signaling data, phosphoproteomics basically, what is phosphorylated. And as time points that shows 0, 15 minutes, 30 minutes, 24 hours. And it turned out everything was over after 5 minutes. So we had before and everything after, but we didn't really see what was going on. So you need to know these timescales in order to also design your experiments in a way that you get the meaningful events. And if you need to do phosphosignaling, then you need 30 seconds, 60 seconds, 120, and that's when everything's done. So that makes it hard because if one of these sets of biomolecules changes within seconds, the other changes within seconds, and the other changes on the order of days, then it becomes just so much harder to integrate that into one meaningful data set. Let's start looking into proteomics a bit more. So as I said, proteomics is the start of proteomes, which is kind of nicely circular. We're really talking about a set of proteins in one sample. That's fundamentally what it is. That can be cell culture, that can be a tissue, primary tissue that you get from a patient, or it can be something that you extract from that. So the sum of all proteins in one sample, and that means we have a defined tissue, we have a defined time point, and we usually want to compare between different samples. So proteomics tries to do two things. It tries to catalog all the proteins. Basically, we want to see what is there at any given time point. That is what we call qualitative proteomics. That's how it all started. In the same way that genomics tried to catalog all the genes that are there, we try to catalog everything that is in one sample. How much is there? And that's quantitative proteomics. So you would like to assign a quantity to. And that can change drastically. I said that already. This is basically the distribution of copy numbers that you get out of an extraction. And you can see that it covers several orders of magnitudes, and that's just from a HeLa cell extract. We simply don't know how far we can go because we know that some of these proteins are present in very low copy numbers, and if that is what's happening for you, then you should be able to go that far down. For example, transcription factors. You don't need a lot of that. Other material like serum albumin in blood, it's just a nuisance. You could argue if you want to have the full proteome, you should know that as well. But for most applications, you just get rid of it in order to actually go down into the things that really matter. And that's another thing you need to know. What are you looking for? Proteome it's not a miracle method where you say, if I have the proteome, I just need to look at the data. You really need to have the question in advance. If you need to know what is it you're looking for. Now, when we look at a protein, proteomics is, yes, please.
What technology is used to reduce the background protein, whatever? Is there something?
Depletion. So you can basically trap some of these proteins and basically use all the rest. So you can deplete serum albumin, for example. Yeah.
So
Just
like
pull it.
thing that you put.
It's in the sample preparation, pre-analytical. Or you can, for example, if you're interested in phosphopeptides, then you trap them on a titanium dioxide column. You do an enrichment. So you can do depletion, or you can do enrichment, or you can do fractionation and then decide which fraction is really interesting to you. But both of these methods aim at reducing the complexity of the sample because the technological limitations of the methods ensure that you can see only a certain range of that, certain dynamic range, certain concentration range. So when we're talking about proteomics, we need to look at proteins first. I hope everyone knows that a protein is just a linear chain of amino acids, usually 20 amino acids with some exceptions that I won't go into. And gives them their function. Form follows function as you. And these amino acids are connected via peptide bonds. So you know that there's a carbonyl group, an amino group. It's basically a simple condensation reaction that connects to a primary amine. You lose water, and fundamentally at these peptide bonds you can break this chain again. So it's rather trivial, and when we read these amino acid side chains from the N terminus to the C terminus, these side chains here define what amino acid that is. If we read them from N to C terminus, that get what's called the sequence of the protein. In proteomics, the typical questions we now have are, what is in there? Protein identity. What is the unique amino acid sequence? And the single source of origin. By that I mean, which protein does that really come from? Which organism does that come from? It might be that you have multiple organisms in your sample. There might be different genes encoding identical amino acid chains. We will get back to this problem on a different scale later on. And different organisms might also encode the same sequence. Then we have splice variants. A gene can arise from different mRNAs, isoforms, protein isoforms that are or are not contained in the database. There are polymorphisms. Many genes occur in allelic variants. Some of them have very high number of alleles. For example, the human major histocompatibility complex has several thousands of alleles that all lead to very different protein sequences. On average, what do you think? What's the average number of isoforms that you have per gene? So when you look in the databases, NextProt, for example, then they average 2.75 isoforms per protein. But with some of these loci, that does not include personal genomic variation. So if anybody in the audience would sequence their genome and compare it to their neighbor, to their left-hand neighbor, how many differences would you observe? Roughly. Yeah. We have many of these that are occurring in intronic regions. So how many different amino acids would that give to 3 million? I mean, we know we share the majority of our genome, but fundamentally we're all very different. That is on the order of tens of thousands of amino acids, 12,000, depends a bit on what you're looking at, that are really different between any two persons in the room. So these would, if you're looking for pathomechanisms in cancer or in a rare disease, then you would really going on the individual amino acid level. But if you're just asking what's the difference between cell culture under state A and state B, then this might not be relevant. But it's a source of variation. And of course, proteins can be post-translationally modified. Where we're talking about phosphorylation, there's acetylation, but there's also oxidation and all sorts of chemical modifications that we can apply. Looking at such a proteome can be quite interesting. However, you've probably seen a picture like this at some point. It's sort of the classic way of representing that caterpillar, and they of course have different proteomes, and they look that sort of why the proteome is because do with genomics or transcriptomics. And one of these things, just to give you an idea, is a paper we wrote a while back. There are problems that cannot be solved with genomics. And that was a paper we wrote on platelet signaling. platelets don't have a nucleus. So there's no point in doing transcriptomics. There's nothing there. What do platelets do? Blood platelets? Is anyone? Yeah, they're responsible for blood clotting, so they are mechanically activated. So if there's sufficient force on the membrane, then these platelets induce a signaling cascade that leads to a change on the surface and basically makes the blood clot and makes them adhere to each other. And apparently you cannot understand this process using transcriptomics or genomics if all that happens is really on the level of phosphorylation, and that's what happens. And you can do that with proteomics, but with none of the other techniques. So there are problems that are really where proteomics is the only method to go to, and there are also questions that can be answered that way only. Let's go one step further and talk a bit about metabolites. Metabolites are intermediates and products of metabolic processes, basically everything that biochemistry can create. So you could argue based on this definition that also proteins are, of course, metabolites, and DNA are metabolites. Of course, true, but traditionally we take these long biopolymers out of that question. And there's a borderline there. 1 amino acid is a metabolite. 2 amino acids, yeah. 10. And if you got 500, you call it a protein. Yeah, so it's not really clear when we include these molecules. You're pretty sure that it's going to be not a metabolite. Other than that, it could be anything: amino acids, lipids, sugars, alkaloids, arbitrary degradation products of everything, things that are taken up with your food, things that these things are turned into. So it can be all of chemistry, and that is what makes metabolomics intrinsically much harder than proteomics, because with proteins, at least you know the chemistry. You know there are 20 amino acids, give or take, and with metabolites it can be almost any chemical structure, and the chemical universe is infinite, fundamentally. But if you have that, then metabolomics can be a very useful tool. Metabolomics is particularly interesting for personalized healthcare because it's readily accessible. We have many samples that we could use, biofluids, urine, blood, that are readily available in the clinic. And if you can do metabolomics right, then you can easily and cheaply also get things that are. And that has been a promise of metabolomics for a long time. If we can find the one biomarker that is going to tell us something about the disease, but reality, again, is a bit more complex. It can also be interesting for molecular epidemiology. When you say, I can study from a biobank, for example, without particular preparation, 1,000 patients in a certain context, the disease context, you can find these metabolome-wide associations. You might have heard of GWAS, of genome-wide association studies. You can do the same with metabolites. What's the problem with metabolites? Why is GWAS a thing and MWAS is not? Well, we're back at the question I had earlier about the dynamics of these different omics levels. And the proteome is also very dynamic. And that means it becomes harder to detect the signal in the variation that we have just based on temporal fluctuation. We can get the patient at the right time point and figure out what is induced by whether they actually walked to the hospital or took a taxi, what is induced by their breakfast, and whether they are hungover from last night. So all these things are factored in, and that makes it a lot harder to really find that. Metabolites and proteins are connected, and the connection is not often not clear to everyone. This is why I'm trying to explain that. You know the citrate cycle from biochemistry, sort of the textbook pathway per se. probably know, citrate is turned into cis-aconitate, isocitrate, and so on. This here is a circle, and that's taken from a database from KEG. And as computer scientists, we have a different take on this citrate cycle. To us, it's what's called a bipartite graph. Bipartite graph means we have nodes of different types, and we have edges in between. And what you can see are these circles here. These are metabolites, citrate, cis-aconitate. And what are these boxes? What is 4.2.1.3? They're enzymes. That's the EC nomenclature. So fundamentally, these boxes are the proteins. So proteins are turning citrate into cis-aconitate. So they are connected somehow. And if you want to understand why the metabolome changes in a certain way together with the proteome, you need to know what are the connections between the two. And that is what these metabolic networks and these metabolic network databases are essential in order to start understanding the data. All right. So much for preliminaries. Now let's start and dig a bit deeper into the technology so we understand where the data comes from. And I get into arguments with some of my students all the time who claim I should talk less about chemistry and biology and more about real algorithms. And that's precisely the subset of my students who run into the most stupid errors afterwards. Because if you don't understand where your data comes from, then having a fancy algorithm basically means that you can shoot yourself in the foot just a lot faster. So in order to understand the data, in order to build the algorithms, you need to understand the data. In order to analyze the data, you need to understand the algorithm. And this goes both ways, and this is why I'm trying to give some of the basics first on where the data comes from, and I'll point out where some of these properties of the data can cause issues later on. So when we're talking about chromatography coupled to mass spectrometry, LC-MS, got a couple of bottles. That's where the solvent is. That's the waste. And we're pumping that through a column that is somewhere here in the oven. And we're trying, at the end of this column, we have one of these machines here, and it's trying to measure mass spectra. So we're coupling two technologies. One is a separation technology, liquid chromatography, and the other is fundamentally just the detector for that, mass spectrometry. Why mass spectrometry? What's so special about mass spectrometry that we would use it for metabolites, for proteins, for lipids, for all these biomolecules? Why not any other analytical technique? Yeah, that is a benefit, but NMR can do that as well. It's sensitivity. Who of you has actually flown in to Boston? Was at the airport. That's why they are using mass spectrometries also at airports, because of the sensitivity, explosive tests there. That's one way to do it. So let's look at these two technologies a little bit and why we're using them specifically. Chromatography basically comes in flavors. Metabolomics only, and both metabolomics and proteomics. And this separation is used to reduce the complexity that contains 50,000 metabolites or proteins, which is not uncommon. Then the mass spectrometer itself is useless on that. So instead, we reduce the complexity of the sample, and then the mass spectrometer to deal with it. And what can it do? Well, the mass spectrometer can identify what you said. We can sequence it. We will see whether we actually doing something completely different. Or in metabolomics, you get something like a fingerprint spectrum. It's kind of an identification. And the second thing is that the signal intensity is proportional to the concentration of the molecule in the sample. So we can actually use the mass spectrometer for quantification as well, and that's an essential point here. So how does it work in proteomics? This here is a gun proteomics workflow. So you start with a sample that contains your proteins, say your cell culture. You extract the proteins, for example, by precipitating them. Then they're usually digested. That's why it's called shotgun proteomics. In the same way that nobody has ever sequenced a genome. Instead, people have only casted the genome to bits together. That's genomics. In the same way, nobody really ever sequences a protein, but instead we digest these proteins with an enzyme, for example, trypsin, and then we look at these individual parts of the protein. Oftentimes, after digestion, the complexity of the sample is still too high, and then you can do peptide fractionation. That could be done using isoelectric focusing, can be done on a gel, off gel. There are different ways to do it. Usually, it's on the order of a dozen fractions or 24 fractions at most. Each of these fractions is then individually run through the HPLC column, and this here is sort of a chromatogram. So individual peptides come off at different times. And then we shoot them into the mass spec, and the mass spec just records one spectrum after the other. So no matter what you do, the data is always going to look like this. Is always going to be a mass spectrum function of time, and the gradients tend to be several hours, and the mass spectrometer can record, well, a couple spectra per second. So you end up with tens of thousands of spectra for complex data sets, or a lot more, depending on how fast you run that. So let's look at these two methods in more detail. Mass spectrometry, by definition, that's an analytical technique identifying type and amount of an analytes present in a sample by measuring abundance and mass to charge ratio. So the name, in a way, is already a misnomer. A mass spectrometer does not measure mass. A mass spectrometer measures mass to charge ratio, and the two things are not the same. The second thing is we also measure abundance, and we will see tomorrow how that relates then back to analyzing the data. Mass spectrometry is related to mass spectrometry. If you meet people, physicists who want to get into mass spectrometry, they always refer to this as mass spectroscopy. It basically means they have no idea what they're talking about. Mass spectrometry can cover a wide range of analytes as a high sensitivity. That is why it's a very versatile technique that can be used for lipids, metabolites, proteins, peptides at the same time. It goes back a long time. It's actually years old. The guy here on the right was the first guy particles with magnetic. Though he didn't call it that. One of the pioneers was Joseph Thomson, a bit later, who built machines, and these sector field mass spectrometers were used for many different things. One of them was actually in a preparative manner, not just measuring, but actually separating isotopes for the Manhattan Project, uranium-235 versus 238. So this fundamental physical principle has been known for a long time, and people have been using it for a long time in physics and then later on in chemistry. And it really took off in the 1950s, 60s when we got so-called traps. So when people started being able to manipulate ions, charged particles using electrostatic magnetic fields. When you look at a modern mass spectrometer, it still has the same components that a mass spectrometer had in the 50s and 60s. It's just the way it is done has changed, but not the fundamental components. And there are fundamentally three of those components. An ion source that takes the analyte and charges it, turns it into ions, into charged particles, because only charged particles can be manipulated by electrostatic magnetic fields. Then you have a mass analyzer, and there you see that this is actually, but you can also separate. So fundamentally, mass spectrometry is also a separation technique. The mass analyzer, their mass. That's all it does. It doesn't count them. It doesn't do anything to them. It just separates them by mass, and then they're recorded by the detector. So if you throw in a sample here that contains different analytes, be it peptides or metabolites, it doesn't really matter, then the ion source turns them into ions, for example, by attaching a proton to them that's positively charged. They are somehow gotten into vacuum, accelerated, and then a mass analyzer would sort them and would figure out that this black guy here is lighter than the red guy, lighter than the blue guy, and pass them on to the detector in some sort of order. And the detector would then determine a quantitative signal that could be, in an ideal world, this would be counting. So we have like 2,000 of these black guys, 900 of the red guys, and 2,900 of the blue guys. And we would return that in the form of this so-called mass spectrum. The mass spectrum is just the count of an ion at a given mass over charge ratio. And you plot that by having the intensity as a function of the mass to charge ratio. That's all there is to the mass spectrum. All the data we're looking at is just these pairs of mass to charge and intensity. It's not more complicated than that. So when we want to combine that, liquid chromatography and mass spectrometry, these so-called hyphenated techniques, LC-MS or HPLC-MS or UPLC-MS, don't be confused about that. It's all the same stuff. It's just marketing. If we combine that, then we can use the mass spectrometer as a detector in chromatography. It can detect hundreds or thousands of compounds in parallel. And HPLC is used to separate them in order to get the complexity down to what the mass spec can handle. And the idea is quite simple. You have different, the mass spectrometer, something went wrong on this slide, I'm sorry. Basically coming off the column at different times, and then mass spec can detect them in order. Now let's look a bit more deeply into mass spectrometry. I said ions are generated by this ion source, and they're then accelerated by electrostatic magnetic fields. That means neutral molecules are unaffected. If you can't get a charge on it, then it won't work. So how about peptides? Are peptides charged? Yes. It depends on modification. What else does it depend on? The pH, right? They are acids and bases. So fundamentally, by changing this pH, and you might know isoelectric focusing, if you change the pH to the right point, you can decharge them. You can also get them into vacuum, and if you have them in vacuum, you can start accelerating them. And there's a very simple fundamental force, the Lorentz force in physics, high school physics. I think some of you might have heard about that. There is a force acting on a charged particle. The force here is F, the charge is Q, and then you have an electrostatic field, and you have a magnetic field E and a magnetic field B, and you have the velocity v that the particle is traveling at. So the force induced by the magnetic field is larger if the particle is going faster. For the electrostatic field, that does not matter. If you put that together with Newton's second law of motion, F equals m times a. You know that from experience, yeah? It requires more force to push heavier stuff, or get to the same acceleration a. We can actually determine the acceleration a of a particle. If we F equals m times a, and if we can determine F, then Q times E plus V times B equals m times a. And this is actually, you see here, m over q, that is mass to charge ratio. So if we can measure the acceleration of a particle, we need to know what charge this particle had in order to determine its mass. And that is why a mass spectrometer cannot really record the mass unless it also knows the charge. So I hope you give me this little bit of physics here. But it's kind of instructive to see where this m over z actually comes from. And it comes really from this very fundamental principle of Lorentz forces and Newton's law of motion. Yes, please.
So how likely would it be for two peptides to have the same mass by charge ratio? Is it technically?
Yeah. Yes. It depends. It depends on fundamental. Let me give you a simple example. You could argue well, the amino acids have different masses, and if you combine them, then they should be different. But you also can consider all possible permutations. Yeah, if you change the order of the same amino acids, then the peptide will have the same mass. And there you run into lots of combinatorics real quick. And you also have to consider the size of the proteome. So people have looked into identifying peptides just from their mass to charge ratio and also retention time. That works to a certain degree, but it's not a very reliable way of identifying peptides.
There are other things that can be used to enhance unique identification.
We will get into that. It is more interesting for metabolites because with metabolites you don't have the sequence. We run into similar issues that we will discuss on Wednesday morning. So now, once we have the ion in the vacuum, we apply an electrostatic field and we just try to push it, accelerate it towards the detector. And clearly, if you have two particles that have different mass, then this acceleration will be different. And you can easily see how that works. If the mass is twice as high, then the acceleration is going to be half the acceleration of the other particle. It's relatively straightforward. And that helps us to sort them, because if you accelerate them, the easiest way to detect them would be to measure the time that it takes until they hit the detector. So you accelerate a bunch of these ions, you measure the time, and if you know that, you can figure out the mass to charge ratio of these particular particles. Now let's go one step back even more and talk about what we really mean by mass. Ultimately, that seems trivial. I mean, it's just the weight of the thing. But it's not that trivial. Atoms, of course, have a mass. That's why we all have a mass. We might want that much mass at some point, but that's how it is. But these elements have different masses depending on the isotope. And I think you're all aware of that. Isotopes are chemical elements that have different number of neutrons. So they have the same atomic number, but different masses. Simplest case is hydrogen, light hydrogen, heavy hydrogen. Either it's one proton or it's a proton. That's roughly twice the mass. Masses are generally given in units of kilogram, not pounds. However, there are different conventions for these various masses, and there are different types of masses depending on what you're really talking about. A mass is the rest mass of an atom in its ground state. Sounds a bit strange. Why should it be heavier when it's faster? But that's just relativity for you. That's the way it goes. They are generally expressed in so-called unified atomic mass units, which correspond to 1/12 of the weight of a particular carbon isotope, C12. And that's 1.660540 times 10 to the minus 27 kilograms. That's this unified atomic mass unit. People also use Dalton, which is a non-SI unit that is equivalent to this unified atomic mass unit. But for various reasons, the International Union for Pure and Applied Chemistry decided to use this term. It's wrong, that's not there, but if you want to get it right in your paper, you put in there these days. Another unit is also the atomic mass unit, AMU. That should also be roughly equivalent to Dalton, but again, it's not the currently supported unit. Now, when we go atoms, this was just atoms. If we go from atoms to molecules, then things can get a bit more complicated still. The mass of a molecule is fundamentally just the sum of the masses of its atoms. Simple. But now we have two different types of masses that we're talking about in mass spectrometry. One is the exact, that is the experimentally determined mass of a molecule, what you really measure. And that comes with a measurement error. The exact mass of a molecule is theoretically calculated, so there's no error attached to it. And that is what you get based on the calculated specific isotope composition. And the molecular weight that you know from chemistry, that is something else entirely. That's the molecular weight. That's the ratio of the molecule's mass to the unified atomic mass unit. But you need to define what you're really talking about. It can be the average molecular weight. It can be the monoisotopic molecular weight. And depending on the context, they can be very different numbers. Note also that these terms are not always used properly in literature. You will find all sorts of units, all sorts of definitions. If you really want to know what's going on, go back to UPAC. The Golden Book has all these definitions. That's where you can look it up. Let's talk a bit about isotopes now. Fundamentally, atom species, same chemical element, different masses, just different number of neutrons, same number of protons and electrons. And for chemistry or for proteomics, it's basically carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur that we care about. Other elements that can occur here and there, selenocysteine, for example, let's not talk about those today. When you look at this table here that contains the various isotopes, their atomic weights, and most importantly their natural abundance, then you can see that for most of these elements we have one predominant isotope, heavy hydrogen, light hydrogen, heavy hydrogen, then light carbon, heavy carbon, C12, C13. It's slightly different when you go to sulfur, where you have isotopes that are 2 mass units, and they actually lead to distinct. Because if you use these distributions, then you end up with a molecule, not just one peak. Finish with 2 more definitions, and then we're done with that. And that's the mass number, the nominal mass, and the exact mass. Mass number is now just the sum of protons and neutrons in a molecular ion. So that is always an integer. The nominal mass is calculated based on the most abundant isotope. Note this is not the lightest, the most abundant isotope, and that causes problems, for example, with bromine. Rounded to the nearest integer. And then you have the exact mass that is calculated by assuming that you have a single isotope, usually the lightest one, for each atom. So if you want to calculate the nominal mass of glycine, C2H5NO2, you take 2 times the nominal mass of carbon, 5 times that of hydrogen, and so on, and you end up with this integer number. That gives you a rough idea of where this should be in a mass spectrum, but of course, by no means this can be used to identify it. The exact mass, however, based on the lightest isotope, that is calculated on the exact numbers here. So now it's 75.032 and so on. But that is not necessarily where you would find glycine in your spectrum. Why not? Because there will be multiple peaks that are associated with glycine. And that is where this term monoisotopic that I used already comes in. Monoisotopic mass of a molecule corresponds to exact for the most of each element of the molecule ion. Just something. That is typically the lightest, but not always. And the mass defect is now the difference between the mass number and the monoisotopic mass, and mass excess is a negative mass defect. So, for example, glycine we have 75.03 monoisotopic mass. It's the same as what I had as exact mass on the other slide, because I'm referring to the exact mass of the lightest isotopes, which tend to be the most abundant isotopes here. But it's not. And the mass excess is then this minus the nominal mass, so we have 0.03 mass excess in this case. Finally, average mass of that's calculated using the average mass of each element weighted by its isotope abundance. Yes, please.
Usually the abundance is like on the order of like 99% of those, or it can be somewhere like 50-50.
Well, when we're talking naturally occurring, then it's just the numbers I had in the table. But you can do isotopic labeling. So if you're doing SILAC, then that's not what you would see. So the average mass is just obtained by weighting this, just the weighted average of what you have. You basically think about how likely is it that I see a carbon that is light, how likely is it to have a carbon that is heavy, and then you just take these probabilities of these relative abundances and you just sum it up. So 98.9% of the carbon are going to be light, 1.1% are going to be heavy. You sum the two up, that's it. That's your average mass. Okay. Accurate mass is experimentally determined. So that's not what you can calculate, but it can be used to actually determine the elemental formula and then determine what it could have been by looking at what are the exact masses of candidates. So accurate mass comes with a known accuracy or relative error, usually given in ppm. As every measurement has an error, accurate mass should come with an error as well. If it doesn't, then you did something wrong or you didn't look closely enough. You need to know the error of your measurement. In particular, as suggested, if you want to identify based on mass to charge ratio, you need to know how often that is off by how much. And we will come back to that. So most mass just have a constant relative mass error or an absolute mass error that increases the measured mass, which is the same thing. It's a heteroscedastic error. You would be able to measure masses with a higher absolute accuracy than these higher masses. That's why you specify it in ppm, in parts per million. Here's an example. If you measure an accurate mass for valine of 117.077, and you have the monoisotopic mass of valine at 117.078979, then you can compute the absolute mass error, which would be 0.1 mass units. And that translates to a relative mass error of 16.9 parts per million. So I know that these are the UPAC terms. That's how it's properly defined. In particular, when you write papers that refer to this mass spectrometric data, you do well to adhere to these terms, because otherwise a reviewer that is not exactly your friend will sort of tear in you over this. You can just look it up. I put in the references where you can find all these definitions. Now let's make use of that. And the most interesting note that we will look into is that of difference. So let's look at a molecule with one carbon atom. This molecule has basically two options for its carbon. It can be a light carbon or it can be heavy carbon. 98.9% of these molecules will be the light variant and the heavy variant. So when you record a mass spectrum of these guys, then 98% of these guys will be the light variant, 1% the heavy variant. So if you have other isotopes, then the same thing holds. Now, what's it going to be like if you have carbon atoms in the same molecule? The odds. So first of all, possible options that we have. How many peaks will there be? The lightest is still the highest. How does it change if we sort of add more carbon atoms to the molecule? Yeah. So will always be the lightest peak the most intense peak? Okay. Well, I see I lost some of you. I'll show you what I mean by that. So these isotopes, if you look at a molecule with 10 carbon atoms, this variant still contains only C12, nothing else. But the other 1 to 10 carbon atoms could be heavier. So on the whole, you could have masses that are also shift by 10 Daltons towards a heavier mass range. So what you measure then, what the spectra look like, are really groups of peaks like this. So instead of having just one peak per molecule, you have the monoisotopic +1, +2, +3, and you see down here it goes into the noise. We have only limited dynamic range. So once we go beyond the signal-to-noise ratio that the mass spectrometer can handle, we're seeing these peaks. They're still, fundamentally, they're still there. If we count enough of these molecules, they're still there. How do we compute them? Well, you can compute them approximately. It turns out that this is a distribution, and happier you go, shift towards the right. So let me give you an example. A molecule of 1,000 Dalton, and you can just assume carbon atoms are there at 1,000, and you compute the probability of the monoisotope, the +3 and so on peak. Yeah. So the lightest, next light, next heavier one, until you run out of atoms at some point. And I only show you the first column. So 55% where all the carbons are light carbons. 30% will have one heavy carbon. Yeah, that's to be read. And that means the spectrum. You have one, the monoisotopic, and then you have these satellite peaks +1, +2, +3 that are lower. If you go to 4,000 Dalton, then it looks differently. Now the odds of being all light are just 9%. And the most likely variant to have 2 of these heavy carbon atoms. And then you see this looks, well, differently. As you go to infinite mass, this actually looks like a normal distribution. If you want to play around with the calculator, there's a link down here. You can toy around with that. You can put in different elements, actually fun to play. You can just take at some point 2 or 3 minutes to play with that. It's easy to understand, but it's get some idea. And particularly quantification, we need to know how these shapes look like in order to collect all the peaks that belong to the same peptide. Unfortunately, what I just told you is complicated by something that's called the isotopic functor. This used not to be a problem, but since we got more and more resolution in the mass spectrometer, tend to see funny stuff like this. So this here is a spectrum at low resolution that you would see monoisotopic +1, +2, +3. And if you zoom in around this guy, then you see there's another peak. And this peak comes from light nitrogen. So shouldn't be the mass difference between N14, N15, and C12, C13 be the same? Obviously not, otherwise there wouldn't be that peak. But what's going on? Where does this come from? Well, that's a difference in mass defect. That's why nuclear power works. Why is that? Because if you put together an atom, you actually do not take the masses of neutrons and protons and electrons and sum it all up. But in order to bring them together, you lose energy. And this is the mass defect, the relativistic mass defect that you get out of fusing atoms. And that's actually, this difference is the energy that you get out of fusion. If you make fusion work, that's what you get. So knowing that can help you interpret these spectra at some point. If you see them with most mass spectra, you will not see them. But if you have high enough resolution, you will start seeing these isotope satellites. Okay, compute isotopes. For simplicity's sake, we will just consider nominal masses and ignore this fine structure. It's not a big problem in most cases, and you do that. Elements and E of the natural isotope. And I is the additional neutrons, so it's 0 for the lightest isotope, for monoisotope, and +1, +2, +3 as I pure as these pi 1 so on until of occurring isotopes, number of naturally occurring isotopes. And now we assume we have a molecule composed of two atoms of element, and we can compute the probability for additional neutrons in this molecule using this rather simple sum. Just add up over the combinations. And that is known as and we can rewrite that operator star. And now it tends to get a bit more complicated combinations. You can see how that can simply introduce powers for arbitrary distributions. If you look at carbon monoxide, so you're basically dealing with two different atoms and two different elements. So these convolutions out over all the different combinations that you have. Just how many, what abundant carbon for an isotopic shift of half. That's, and you can easily elements. This sum, and then you get all the combinations, you sum up over them. It's really straightforward. Okay. Spectrometry and metabolomics is a very brief introduction. Part of the lecture. Okay. Now, software that we're working with, and that is OpenMS and also KNIME. And we will talk a bit about workflows, and I'll introduce the concepts behind OpenMS and KNIME. And then we will start with workflow. So, this project 15 years ago, and one of the things that has happened since is that the amount of data that we have has grown exponentially. This here is a chart from EBI, the European Bioinformatics Institute. Again, we have a log scale here, and that's the size of the data in bytes. And this here is the time, the year when the data was deposited. And again, Moore's law are these dashed lines. That's exponential growth. And there are different types, EGA, ENA. These are nucleotide archives. And you see how nucleotide data is growing at the same rate as roughly these dashed lines. So the data is growing exponentially. Proteomics, that's bright, is growing faster than exponentially. It's crossing these lines. And metabolomics, that's what they're really scared about, because that is growing a lot faster than exponential. And why am I showing that? Well, if we believe that analyzing this data as it gets larger, then we're in trouble at this point, and also storing this data becomes a bit of a hassle. So Proteomics, what we're really trying to do here is what everyone calls big data. That's another buzzword that you can put in your grants. You're doing big data on omics in order to get systems biology right. And if you do AI on top of it, then it should be good to get that grant through. Talking about huge data sets, often hundreds of gigabytes, terabytes, heterogeneous, and we want to integrate them. And you might have followed this debate. It's harder to extract good science from large data sets. But it's something that is essential, and you need to build the skills to do that. We want to extract knowledge out of this data. You don't know, most people don't do proteomics just for the fun of doing proteomics. That's one reason why you might do it. But often you have a question that you want to ask. And now the art is really, if you generate 1 terabyte of data or 10, how can you still extract a meaningful statement, a biological hypothesis or a statement that you would have gotten out of a gel otherwise, where it's simple? How can you extract that out of a terabyte of data? And the problem here is that it becomes harder to do that. There is a debate in the literature has publishing a few things there on reproducibility crisis. You might have heard of that. And in parts that is also associated with the reproducibility of data and data analysis. If you want to pull out data out of 1 terabyte, the statement, the following 10 proteins are differentially regulated, then you need to be able to do the same thing also a year later. Now, you might, if the data is the same, I should get the same results, and in theory that would be true. But in theory, you could also argue you would get the same once you repeat the experiment. But both statements are not true. It's exceedingly hard to get exactly the same analysis. You don't do exactly the same it was done, exactly the same way it was done in the first place. That's what we're talking about, reproducibility. A second aspect is actually throughput. If you use high throughput methods, you're generating data at an unprecedented speed, and analyzing 1 sample is usually not a big deal. You just down-click through the spectra, and you give your PI like a summary in 10 lines what you saw there. But analyzing 100 samples can be painful, and at 1,000 samples anymore. So compute and analyzing a data set to death. So we really want to go for automation, and that is where the term pipelines or workflows come in. Now, you know pipelines from these big-ass oil pipes that you pump water through, and if you go to the dictionary, then you find this definition, of course, a long pipe, typically underground for conveying oil gas over long distances, but we are, of course, interested in the second definition. In computing, pipeline means a linear sequence of specialized modules used for pipelining, which is embarrassingly circular, but you get the idea. You modules just with, as with pipelines that tend to leak as well, not as much data will fall out at the end, but this is what we're looking for. Now, a workflow, the definition of a workflow is a sequence of industrial, administrative, or other processes through which a piece of work passes from initiation to completion. That's just a definition. It just something comes out. General term, and we use the term scientific. Meaningful data, hopefully. And bioinformatics to be faced comes to us. It's a hard disk on our desk and says, well, you're a bioinformatician. My people couldn't make any sense of this data. Your turn. And they believe that magic pipeline somewhere hidden in the basement, and it always has to be an Excel sheet. Then I'm trying KNIME. There are other tools, and that make up the nodes of this workflow, and now data is flowing through these. And what goes into one tool comes out of one tool, goes into the next tool. So you chain them up, and we try that in a hands-on, really putting together work. But you can also plug in other tools like R. Once you have them, you can make workflows out of that. To take a break here, and then